So good afternoon, good evening, everybody. We are starting the last session. And before starting, I want first of all to thank all the organizers under the lead of Biat, all speakers and all participants. I think it was an amazing, very special conference. And we are waiting to see the future and the next conferences. And I want to present you and invite to speak the last speaker of today, Professor Ehud Gazit from Tel Aviv University, and the founding director of BLAVATNIK Center for Drug Development, and also he served at Tel Aviv University for many years and was the vice president for research and development and chairman of the board of directors in Ramat, which is a translational center. He had a lot of prizes, a lot of rewarded, was responsible on, had 350 peer reviews publication, more than 100 patents, is a fellow of the Royal Society of Chemistry, member of Inborn, and a foreign fellow of the National Academy of India. I mean, he's done everything and is a very rewarded scientist, and he will go and talk with us on self-assembly in inborn error of metabolism disorder, new therapeutic paths for rare disease. Udi, please.
Thank you very much, Mira. It's a pleasure to be here. I would like to thank the organizer for the opportunity to speak. This was a wonderful meeting, and we heard a lot about nanoscience, nanostructures, self-assembly, and today I would like to provide a relatively fresh view about self-assembly, which is related to diseases that traditionally were not regarded as those related to this phenomena. But let me start with something that is well known for many years. Self-assembly of amyloid fibers is associated with human disorder, major human disorders, including Alzheimer's disease, Parkinson's disease, type 2 diabetes, ALS, Huntington's disease, many disorders in which, in all cases, you could see the formation of self-assembled nanoscale structures with a diameter of about 6 to 10 nanometer. They are well ordered one. You can see the diffraction pattern using X-ray fiber diffraction, but predominantly beta sheet secondary structures. And very interesting is that fibers from different origin show remarkable biophysical and autostructural properties, the properties that I mentioned, as well as others. So, but in spite of that, there is no simple homology between amyloid forming proteins and polypeptides. So for many years, the key question was, what is the molecular basis for the recognition and self-assembly process? And now our approach for more than 20 years by now was to try to identify short recognition module that can form these amyloid structures. We demonstrated that short peptides can do this process, systematically analyze a peptide fragment to pinpoint the residues that play a role in the molecular recognition and self-assembly process. And based on the mechanistic insights, we identify novel fragments, short as tetrapeptide, that can form amyloid-like nanofibers and develop new ways to control this process. I spoke about it in many occasions, including previous cleanup conferences. So this is work that done many years ago. This is summarized a lot of work, one paper in Science, the other in Israeli Journal of Chemistry. This was our ability to identify the ability of dipeptide, diphenylalanine, to to form ordered assemblies, amyloid assemblies, the same one as you can see in the brain and the of individuals with Alzheimer's disease, Parkinson's disease, and many other very devastating disorders. And again, this is a scheme that represents a lot of work, both from the intellectual point of view, but also from the practical, moving in this case, in the case of beta amyloid, from a 42 amino acid polypeptide that can form these deposits in the brains of Alzheimer's disease patients, to a hexapeptide, and then to a dipeptide that can form these structures. So we were able, with a lot of very systematic reductionist approach, to pinpoint the two residues that are critical for the self-assembly and the formation of nanostructures that are involved in diseases such as Alzheimer's disease. Although this work, as I said, was done more 17 years ago, it is always full of surprises. And this is just something that was published a few months ago in Angewandte Chemie. We were able to solve the crystal structure of these amyloid-forming dipeptides. And we could see the organization of the peptides in the presence and in the absence of inhibitors of the process. And we could truly provide mechanistic insight on the way that the inhibitors affect the solid state arrangement, the organization of these peptides. So you can see in the left side, the organization, the crystal structure organization in the absence of inhibitors, and in the right side with the inhibitor. So it's moving from a beta sheet, a classical beta sheet structure, into a helical one. And thus we can understand the effect of such therapeutic compounds on the process of amyloid formation. You can see here really the full crystal packing in both cases. And this is also allow us to use computational tools to understand the way in which the inhibitors interact with the molecules using molecular modeling. Not only that we could see that the structures formed by the peptides have the biological properties that are associated with these diseases, we also discovered that they have very interesting physical properties. We realized that structures formed by the dipeptides that can form amyloid fibers have very strong mechanical rigidity. You can see here AFM imitation experiment in which we could see the mechanical rigidity of just self-assembly of peptides in the amyloid form that has Young's modulus, which means the stiffness of the structure, similar to what we see with the bone or teeth. Later on, so this was published in 2005, in 2011, Thomas Knowles from Cambridge University and Marcus Buehler from the other Cambridge and from MIT showed the same mechanical properties that is a characteristic of amyloid materials that is made by proteins and peptides. So we proved that we can see not only the biological property, the organization, the beta sheet structure that is similar to what we see with the full-length protein, but also physical properties like mechanics. We could see also physical properties like luminescence. So again, more than a decade ago, we could see luminescence with the structures made by the minimalistic peptide, dipeptide. And a few years later, the group of the late Christopher Dobson, together with Gabriel and Clemens Kaminsky from Cambridge University, who demonstrate optical properties, blue luminescence based on quantum confinement of peptide nanostructures with emission at 450 nanometers, also by protein-based amyloids. Other properties that we could see, physical properties, are piezoelectricity and others. Over the years, we study all kinds of different analogs of the diphenylalanine, diphenylglycine, various derivatives of the structures, but most of them, as you could see here, those that could form amyloid-like structure, fibrillar hydrogel fibers, they all contains the phenylalanine motif. And we could see that for peptides to form amyloid-like structures, you can have very short peptides. They can form ordered assemblies. They have, as I mentioned, the characteristic, just as you see in the brains of Alzheimer's disease patients or Parkinson's disease patients, or in the pancreas of type 2 diabetes patients. They have similar mechanical properties, they have similar optical properties, they have piezoelectric properties, and we could determine that in order to have the structures, you need aromatic residue, mostly phenylalanine, and a peptide bond. But something that is started as a control was really changing all the way that we think about the formation of amyloid and amyloid-like nanostructures. So we could see I will go back to some of our list of various peptides that can form amyloid-like structures. We see diphenylalanine, we see triphenylalanine, we see F-MOC-phenylalanine, we see arginine, glycine, aspartic acid phenylalanine, we see phenylalanine proline, we see naphthylalanine, triphenylalanine. So we ask ourselves whether could a single amino acid form also amyloid-like structures? The answer should have been no, because until a decade ago, we were under the impression that you must have a amide bond in order to form this assembly. Quite to our surprise, this is a paper published in Nature Chemical Biology exactly a decade ago. Quite to our surprise, we could see that single phenylalanine, just the amino acid, could form things that look like amyloid, as you can see in panel A. They bind Congo red and show typical green apple birefringence, as you see with amyloids. They bind THC, another indicative dye, as you see with amyloids. And this was really quite remarkable. Yet, we could realize that the concentrations in which we could see the formation of amyloid-like structures by a single amino acid were quite high. While we could see the formation of structures by a peptide with concentration in the range of micromolars or tens of micromolars, here we needed concentration of millimolars, so thousands of micromolars, which on the first thought appeared to be something completely non-physiological, non-relevant. But when we scratched our heads and realized that there are conditions in which you have in the body, in the human body, phenylalanine concentration indeed of a millimolar. This is in a case that is known as phenylketonuria. Phenylketonuria, it is a genetic disorder in which individual lack the enzyme that converts phenylalanine into a tyrosine. Due to this lack of enzyme, inactivity of the enzyme, the individuals accumulate phenylalanine, they cannot produce tyrosine, and the concentration of phenylalanine in their body, in the blood and the brain, is indeed in the millimolar range. Actually, the clinical definition of PKU is 1.2 millimolar. For all of you to be relate to PKU, maybe some of you did not hear about this before. As you know, every newborn, just few hours after his or her birth, are being pinching the toe to draw some blood and test for genetic disorders. The one that is being tested is PKU, phenylketonuria, because if the individual is affected with this disease, they have to go through a very severe diet in order to reduce phenylalanine concentration from the millimolar range to a micromolar range. If not, they have a significant neurological disorders. So we could realize that the structure are being formed in concentrations that are relevant to the disease. But we still had to realize whether, like in the amyloid case, we see any toxicity with these structures. And indeed, as you can see in panel A, when phenylalanine concentrations of fibers are higher than 1 millimolar, we could see a significant reduction in cell viability. In the past, people were thought that the reason for cytotoxic effect in these diseases is just by the accumulation of the amino acid, imbalance of amino acid with regard to transporter and other issues. But we had an excellent control. We added alanine. Alanine, it's another amino acid. But what is the difference? Alanine cannot form amyloid structures. So you can have even 15 millimolar of alanine, you don't see any reduction in cell viability. Only when you have above 1.1 millimolar of phenylalanine fibers, you see the cytotoxicity. So we could see the formation of things that look like amyloid and they are toxic just as a protein and polypeptide amyloid by a single amino acid. It was completely unheard of before this work. More than that, we could raise antibodies toward these fibers, and using the antibodies, we could deplete the toxicity of the fibers as we could see it. So the true story is that we find it, we did also other controls, which I don't have the time to go into it. We send it to an elite journal and they say, well, it's very nice, maybe convincing, but please show it in human patients. So luckily I had a very excellent and stubborn PhD student. She is now already a professor by her own at Tel Aviv University, Adler Abramovich. We went all around and found a brain bank in the Netherlands of individuals, of course, post-mortem of brain slices of PKU, and we could show that the position of the amino acid nanostructures within the brain of the patients was co-stained with the antibodies and with Congo red, this dye that binds a protein and polypeptide amyloids. Later on, since it's so simple to work with a single amino acid, that's all, immediately many groups around the world started to work on this. This is a work that I really like from the group of Michael Bowers at Santa Barbara. Try to understand how come that the single amino acid structure are so toxic. So what he could see that in a neutral pH, there's electrostatic interaction between the negative and positively charged termini of the structures. They form the assembly, as you can see on the left side. So what you have a structure which is really nicely bound, but you have hydrophobic aromatic amino acids that are projected towards the ocean. So it immediately interact with hydrophobic environments such as membranes and does membrane destabilization. And indeed, we can see this case. So if you remember the reductionist scheme that we had for many years, we now reduced it also to a single amino acid, as you could see here. However, phenylalanine is only one type of metabolite that is accumulate in inborn nerve metabolism disorders. Unfortunately, many genetic disorders, many metabolites are being accumulated, and we, in a very systematic way, went through the list, checked experimentally all kinds of different metabolites, and realized that not only phenylalanine, but also other metabolites like adenine, orotic acid, cysteine, tyrosine, uracil, and now other groups have discovered that also other metabolites can form things that look like amyloid, bind THT like amyloid, bind Congo red like amyloid. So we really established that that non-peptide and protein entities, metabolites that accumulate in inborn nerve metabolism disorders, genetic disorders that are affected the newborns, that form amyloid-like structures. Recently, this is a collaboration with Daniel Siegel from our department. We could see that not only classical metabolites, but also sphingolipids could form amyloid-like structures. Those of you who work with amyloid, you can see the beautiful nanostructures that looks just identical to what we see with beta amyloid in the Alzheimer disease, with the phenylalanine in the PKU, with alpha-synuclein with the Parkinson's disease. Also, these form typical amyloid fibers, as you could see by electron microscopy and also atomic force microscopy, and they have fluorescence properties, as we see with the amyloids. And later on, this is another collaboration that we had with a group in Rambam. We could see the formation of amyloid structures with oxalate. This is an interesting story. We could see pathological phenomena with children with hyperoxaluria. They have high concentration of oxalate, but do not form crystals in the retina. We could see using electroretinogram, ERG, dysfunction of the electric signal in the retina. We looked for structures that could be formed by the oxalate. We could see that oxalate also could form fibrillar-like structures. We injected to the eyes of rats, and we could see in the rats exactly the same pattern as you could see with the patients. So we moved from the bedside to the bench and proved to get the same clinical manifestation as you can see with the patients. So just you can see, I'm moving back. We see with different metabolites that are involved in inborn nerve metabolism. We see it with sphingolipids, and we see it with oxalate, just exactly as you see with the patients, as you can see here. And for us, this is we call it the extension of the genetic amyloid hypothesis. These are a few reviews and works that we published about it. And we see by now that indeed various metabolites, non-protein entities form amyloid-like structures. So this is the properties. This is the biology properties. But if you remember, I was speaking also about physical properties, as we see with peptide amyloids and protein amyloids by us and others. So also in the case of the metabolites, and again, this is much more recent, we could see with structures made by adenine, tryptophan, tyrosine, phenylalanine, just the same phenomena as we could see with the peptides, as the Cambridge group could see with the proteins. So metabolites, peptides, proteins, just the same optical properties. And recently we could really prove that there were some suggestions that it might be real, but rather relates to some contamination or oxidation, we could see that we can switch on and off this optical properties, but I don't have the time to go into it. I was speaking also about mechanics. This is the peptides. Few years later, the proteins, and just less than 2 years ago, we could see the same thing also with the fibers made by the metabolites. So biological properties, optical properties, mechanical properties, we see just the same phenomena also with the metabolites, and also piezoelectricity, not by us, another group in Ireland, which are now our collaborator, could see it in the, also with the amino acid structures. So this is a list of the various properties that we could see with the structures that are made by metabolites and so also protein. Shared properties of protein and metabolite amyloid-like assemblies. Kinetics, dye binding, morphology, fluorescence, membrane interaction, neurogenicity, and others. So it appears to be just similar properties. Also effect of chemical chaperones, could see in the inhibition of the process, very similar to what we see with proteins by EGCG or tannic acid and others. We have yeast models to understand this. So it is well established, like just as we see with the proteins and peptides. I don't have a lot of, just finally, this is really published very short time ago. We could see that also in terms of solubility and thermodynamics, this is just a continuum. When you have single phenylalanine, diphenylalanine, triphenylalanine, this is just the same, and it provides a full continuum. And what I try to convince you this afternoon, evening, is that this is just one phenomena that you could see with either proteins or peptides or metabolites, and we bring along really a new concept in the understanding of inborn error of metabolism by self-assembly and organization of the structures. In the last few minutes, I want to share with you a closure of the cycle, how we bring it back to the relationship to the diseases which I started to speak about in the beginning, like Alzheimer's disease, Parkinson's disease, and other disorders. So one thing that was known, it was discovered well before our times, is that in some of the diseases, you can see abnormal high levels of metabolites. But there wasn't any mechanistic way to relate those phenomena to any physiological understanding. So what we could see, some noise, what we suggested, that it might be that metabolite structures could form seeds that actually induce protein aggregation and then seeding of proteins into amyloid structures by the metabolite amyloid may clarify this unexplained epidemiological association. And indeed, we could see in the case of Parkinson's disease, it was well established that high concentration of quinylonic acid, which is on the biosynthesis pathway from tryptophan, are related to high occurrence of Parkinson's disease. Indeed, we could see that also quinylonic acid, as you see in panel A, form typical amyloid fibers. They are toxic, as you see in panel B. They have crystalline structure, as you see in C, and this is the crystal structure as you solve it. Just a beta sheet structure, as you see with the proteins and peptides. And you can see here the magnification of the electron micrograph. For those of you working on amyloid, self-assembled amyloid, it would look exactly like the amyloids that you could see in brains of Alzheimer's disease patients. But we could see that this structure formed by quinolinic acid could seed the aggregation of beta-synuclein that forms amyloid in the case of Parkinson's disease. Also, in the case of the amyloids forming by the sphingolipids, there was indication by association or statistical, epidemiological association between sphingolipid accumulation in Gaucher disease and Parkinson's. As we could see, I showed you already this slide, that it can form assemblies and it can induce the association of alpha-synuclein in the case of these structures. Finally, a work published less than a year ago, we could see homocysteine is another marker that is associated with Alzheimer's disease. We could see the structures formed by homocysteine and also yeast model, but I think that this is a good slide to conclude. This is from mice models of Alzheimer's disease, 5-10x models, which you could see the staining, co-staining of 6-2E antibodies against beta-amyloid aggregates and against alpha-synuclein. And you can see the merging, the co-aggregation of homocysteine and beta-amyloid in the brain of AD model mice. And you could see the really the correlation between the crosstalk between metabolites and proteins. So speaking about all of this, it goes back always to what we are discussing for more than 2 decades. It's simplicity is the ultimate sophistication. Leonardo da Vinci mentioned it already hundreds of years ago. Of course, this work was supported by many groups, including the ERC and Israeli source, American source, European source, and others. This is the group over the years. I mentioned some of the names, some of them were already professors. We already have 13 professors out of the lab, and also many people in the industry in top positions. I moved fast to be exactly on time, although we started a bit later. And if someone is interested in the original work, you can find it on my website. So thank you very much.
So thank you very, very much, Udi, for the broad span and, you know, all this fascinating research. And I just wish you to work forward, to move forward very fast. We need it. Really, I think it's a wonderful closure to a wonderful, very special, very innovative, very interesting conference. And I didn't find any question yet. I have some, but I wouldn't bother you now with that. We will move to the room and then maybe we will ask, we will have more questions. But before leaving and before summarizing everything, I want to say my personal great thanks to Biat and to provide him the opportunity to summarize this wonderful conference. I just want to say one thing, that in my lecture, Biat, I mentioned the beginning in Berlin because of the opening remark of Patrick Uziker 2 days ago. And I said that you told me your vision. And you see, dreams come true and sometimes even better. So just go on and good luck.
Dream
Yeah,
on,
the floor is yours.
Dream on, dear Mira. Dream on. I will dream with you. Udi, you have been in Basel and it was live. And now you are virtual here. And it's so great that you have accepted to do this. It's a real honor. And Peter Kalish and you will now be in the debate room where we will go. But I first want to say to all people that have attended and are still maybe online, that this was for us a good conference and we tried to make it smooth And I think there were practically no hurdles. What astonished me is that sometimes people did not ask or dare to ask questions. I don't know. It seems not to be very popular to tap in questions. I don't know. But the debates that then turned out in the room where nothing is recorded always seem to make a lot of sense, and it looks like people love it. Also, when then scientists, among other scientists in groups, discuss further. So I hope that it will be possible to get Clinam into reality again, and that this hopefully will be possible in the next years. Thank you all, and we wish you well.